So good morning, everybody. This is Cristianne and Rijcken. I'm very pleased to share this session. Beat actually gave the session a rather provocative title, but I must say that after I've seen the focus of the 6 different talks, I'm very eager to actually learn on the different innovative approaches, including also their translational results obtained so far. Then I have myself the pleasure to share actually an update about Cristal Therapeutics. So my name is Cristianne Rijcken, and I like to give an update about the so-called CRIPAC platform that stands for cross-linked polymeric micelles. So clearly, you all do know the EPR, the enhanced permeability and retention phenomena, and clearly what it goes along is making sure the tumor is actively targeted and you have less exposure to the healthy tissue, so really shifting the balance. And by taking a look at the biology, we've kind of taken that as the design criteria to make these CRIPAC nanoparticles. So that means that the design criteria is that the drug should be initially stably entrapped, should have a long circulation, preferential uptake in the tumor, and there only selectively release of the drug that was entrapped over time. And as you may have seen this before, this is how the CRIPAC composition is composed. Drug is covalently linked to the polymeric system, so we have not a physical, let's say, reversible system, but rather the nanoparticle as a co-cross-link structure. But clearly over time, the native drug is being released as a result of cleavage of the drug linker bond. And actually what we've developed over time is that we really have a monodispersed system, namely in this case where we use size exclusion MALS UV analysis. We nicely demonstrate that the drug is really overlapping with the nanoparticle, so indicative that the drug is really in the nanoparticulate system. And if we administer such a composition to mice, intravenous administration to mice, and then we compare the blood profile of the API versus the native drug that was also administered intravenously, then you really see a significant shift of the pharmacokinetic profile. I like what we aim. We really see also co-crossing particles initially stable and really having this nice long circulation. Not only is the co-crosslink essential, but we also demonstrated a nice collaboration with University of Mainz, that actually these particles that we've developed have a really negligible protein corona. And that was nicely demonstrated by AA4, particularly follow-up analysis by DLS and also even proteomic analysis. But all in all, we hardly had any protein absorption, so another reason for the really nice long circulation. And clearly, it's also about what is the actual uptake into the various organs. And there we've done a study together with Rijcken, where we used size 7 label particles, so really monitoring the fluorescent label. And if you then take a look over time to the various organs, then we saw that predominantly after 2 days, the tumor was enriched by the nanoparticles, and that the main organs of eliminations are known liver and spleen. But as you can see, to a lesser extent, they accumulate these particles. The advantage clearly of size 7 is you can really monitor it non-invasively, but after the study, we also excised the tumor tissue and we monitored locally where the nanoparticles were residing. And what we could measure is that after 2 days, we had 2/3 still extracellularly, and if nanoparticles were taking up, they were predominantly taking up by more macrophagic systems. So we really have a good understanding what the detailed macro and micro biodistribution is. So this system is what we've developed, and now I'd like to guide you through the clinical data that we've obtained with the lead product, so-called CPC634, and that stands for docetaxel, which is entrapped to like the same approach. So linker plus polymer, creating the 65 size nanoparticles. We successfully upscaled the synthesis together with Ardena, so that's really clinical scale. And we by now also demonstrate that we nicely can make freeze-dried product, although the clinical batches were a frozen solution, which had a stability of more than 5 years. So what we've done is actually expose almost 100 patients in 4 different clinical trials. I would say more standard phase 1 dose escalation, and then phase 2 small scale efficacy evaluation. We clearly also want to learn more on the platform, and that's where the so-called CRITEX and PICCOLO study come into play, where we monitored really tumor uptake in the patient setting. So the phase 1 trial that was already done a couple of years ago. But just to refresh your mind, the key outcome here was that also in the clinical setting, we really saw a long circulation of the nanoparticles as compared to Taxotere. So a half-life of roughly 33 hours. But even more, we saw also some efficacy signals, including some partial responses and particularly also induced biomarker reduction. Then from the safety perspective, It's good to mention that Taxotere, so the native drug itself, does already have as a dose-limiting toxicity the so-called neutropenia, which really can be life-threatening. We did not see that for CPC634, but we did see another adverse event which was known to Taxotere, namely skin toxicity. And that was particularly upon repeated administration. But what we could also demonstrate if we gave a lower dose plus dexamethasone pre-treatment, then the skin toxicity was well manageable. It was absolutely not grade 3 or grade 4. So as said, that was a more traditional dose escalation, but the most exciting study for me was the so-called Rijcken study, where patients were given either Taxotere, so the conventional drug, or CPC634. So vice versa, so each patient was his own control, and then we took biopsies of the tumor tissue over time, so at different time points as indicated here. And besides that, we also took skin biopsies just to further understand the mechanism for the observation of the skin toxicity. And clearly this is a very brief summary for you to the publication if you're more interested, but the key conclusion was that within the same study, so again with each patient being its own control, we nicely could again demonstrate the much more prolonged circulation of the nanoparticles as compared to Taxotere, a really significant drop of the neutropenia from 75% to less than 5%, and the most appealing clearly was the fourfold higher uptake of the nanoparticle as compared to the Taxotere. So really indicative that the long circulation really resulted in well uptake in tumor tissue. But clearly this was done via tumor biopsy, so needle puncture, operating room, so not really very beneficial for a patient and particularly not applicable larger scale. So actually in parallel, what we've also developed so-called Piccolo trial, where the nanoparticles were actually decorated with a zirconium label, zirconium which has a very long decay half-life. And then the setup was as follows. Initially, we screened the patients with FDGs to see the active tumor lesions. Then we gave a low dose, what we call a tracer or a diagnostic dose only. And then thereafter, we gave actually the therapeutic dose, but that was also supplemented with this radioactively labeled material. And what we can nicely demonstrate is here non-invasively, again, the long circulation of the nanoparticles in the circulation. And then thereafter, we could also monitor the organ accumulation. And as you see in the right plot, that's the percentage injected activity versus kilogram of the body. Again, the known organs of spleen and liver. But the most appealing to me is that actually the therapeutic and the diagnostic dose, they actually had almost similar levels. So really indicative that the diagnostic dose with a 100-fold amount is really very predictive for the actual therapeutic dose level. But clearly, these are all the, let's say, normal organs, but at the end of the day, it's about the tumor uptake. And what we could demonstrate in this study is that we did see 44% of the evaluable tumor lesions taking up the PET signal. So indicative that the nanoparticles they are taking up in some of the tumor lesions, but clearly this also confirms the heterogeneity that is known. But what we've found very appealing is that also here, the diagnostic dose was very predictive for the actual therapeutic dose. So for us, this was a true, let's say, confirmation at this diagnostic dose. So not exposing patient to any therapeutic and potential toxic dose level, the low dose can really be predictive for the future. So you could use this as development companion diagnostic. And this is just recently accepted for publication in advanced materials. Then last, the phase 2 study, which is like formally discussed the evaluation of an objective response rate, in this case, in ovarian cancer patients, advanced metastatic disease, I should say. And this is in a 2-step approach where in total 25 patients were treated. And then clearly it's all about the therapeutic index. So what is happening on the safety side and then balancing that with the efficacy side. So from the safety perspective, we could nicely, again, confirm the improved safety profile. So the neutropenia, again, dropped significantly. The skin toxicity that we saw in the phase 1 was not a meaningful side effect, meaning it was well tolerable with the dexamethasone pre-treatment. And we actually did not find any new toxicities. From the efficacy perspective, we did see some initial signs, but unfortunately, we did actually see too much progression in the patients, as said, advanced ovarian cancer patients with already quite some pre-treatments. And that means that we actually had some additional restriction in the so-called stage 2, whereby we could extend the progression-free survival, but all in all, not a really significant breakthrough. And actually, we think the patients were unfortunately already too far advanced. So that's for us, the key conclusion is actually that we have a really very nice safety profile of the CPC634 product. We have seen observed a significantly improved tumor uptake, including that we can monitor this non-invasively. But in the phase 2 trial, we saw a profile alike Taxotere in other advanced diseases. So what we've said ourselves is for the future, it would be better if we evaluate this product in other patients which are not that aggressive and where a patient may benefit more from the improved safety profile, such as, for example, prostate or lung cancer. But clearly, overall, this also validates the CRIPEC platform and where we are very much collaborating with other companies in co-development in both oncology as well as beyond. And actually, the key lessons learned, and that's kind of the scope towards the future, what we've learned is we do have really nice uptake in the clinical setting. But clearly, it also all goes about explicit exposure in the tumor setting or in other disease tissue. So we're working on different type of linkers to really have optimal release depending on the API as well as the underlying disease. Besides that, we're also working on different type of modalities that we entrap in the nanoparticles. And then this is a very brief example of sRNA being entrapped, whereby literally the entrapment results also in the prolonged circulation in the bloodstream as compared to the naked sRNA. And that translated into an improved tumor uptake. But also we're working on modifying the surface of the nanoparticles. And here too, we had a serendipitous finding, namely that the 7-membered ring that we developed ourselves is a new copper-free click reagent, which we use ourselves to conjugate to azides present on the surface of the nanoparticles, either for imaging, as I've shown you the zirconium labeling or size 7, but also that we can use it for vaccine application. As this, what we call clicker agent is actually really very beneficial for its small size, very hydrophilic, but particularly also very high reactivity. This really is a new enabling agent that we think has also an applicability in the general field of bioconjugates, so not only in nanoparticles. So here we are working on different type of modalities, and more specifically what we've done a head-to-head comparison of making antibody drug conjugates where, let's say, the classical click reagent is so far DBCO, quite hydrophobic molecule, versus in this case where we use our clicker reagent. Then we nicely could demonstrate already a significant shift in the hydrophobicity. So that means that we can generate much more hydrophilic ADCs, which is way more beneficial from the biological exposure. And this is clearly work that has just started. So I'm looking forward to present data in the near future. But all in all, I think we have developed different type of platforms to address the current product challenges that are listed here, such as PK, toxicity, and delivery issues. And all in all, I think I have to stop here. So I'd like to thank all the patients who participated and all the collaborators and finances. And with that, in case you have any question, I'd like to ask you to keep that for the session at the end. Then I presented something about polymeric nanoparticles, but I then have the pleasure to give the floor to Stefan Halp there, who will present about liposomes. And more specifically, I'm very interested to learn about your rather impressive clinical pipeline that you've developed in recent years. I do have seen quite some recent LinkedIn updates, so I'm very curious to learn about the progress. Stefan, the floor is yours.
Thank you very much, Cristianne. So it's my pleasure to present to you our latest insights in the project aiming to develop targeted liposomal doxorubicin, which is an acronym for the name of the product, TALI-DOX. I'll speak a bit about the molecules that went into the mix, so the biophysics, some preclinics, and then mostly clinical evidence that we have so far, and the road to markets. So I want to start with a question: do we really need new liposomal doxorubicin? I get that a lot while working on it, and I think it's an interesting question. I want to point out that at the point where I started in Omedica and I finished my PhD was around 2012, I came across this paper that I hope most of you know by Scannell and colleagues, indicating that the development costs for new drugs are increasing basically every year to a significant amount. And I was puzzled reading that, while technologies such as DNA sequencing and nanotechnology should have actually maybe cost the opposite. So I made a quick research to update that chart. Today, we're clearly above 1 billion in cumulative costs in R&D to give rise to a new drug. If you imagine how much money went into RNA-based nanomedicines, 20 years of development that those particles took. These are gigantic spendings, and I think it requires innovation from all areas to come together to resolve this issue. And so I think, in brief, that nanomedicine actually can resolve at least part of it by taking medicines that are very well established, have a very good safety and efficacy profile themselves already. I, for example, like this World Health Organization's list of essential medicines, of which doxorubicin is part of. And I think there is, for example, one issue in conception, that we don't really make existing drugs better. I see Cristianne working on docetaxel, but the amount of people that make efforts to actually make existing drugs better and not only copying them and selling them at a lower price, such as generics, is, I think, not yet that where it should be to resolve actually a lot of burden that these drugs are causing, such as adverse effects that can be very well upgraded by the use of sophisticated nanosystems. And I think the beauty of it is actually something like the network effect, where you can upgrade many APIs using the same nanosystem. So in brief, there were 45 years from the development of clinical doxorubicin, so adriblastine, to Doxil. And then again, it was like 27 years to come up for us with Talidox. And I hope, at least there is a reduction in time span, which is required to come up with new nano formulas. And I hope that we can definitely shorten that period again to maybe 14, 7 or 3.5 years soon thereafter, by having vertical integration. So coming down to the product itself, I think there are 3 key points that must be addressed with a new nanomedicine or a platform that is generating these. It should penetrate well into the tumor, of course, and release drug there. It should activate the tumor microenvironment in a way that it becomes less immunosuppressive, so the immune system can launch a full attack. And it should be obviously very well tolerated to have a high life quality of the patients. So biophysicochemical rationale of Talidox is manifold. The most obvious parameter is obviously size. It's very small, 35 nanometers in average diameter as measured by cryo electron microscopy. So having smaller particles is very beneficial, for example, to extravasate in tumor tissues, such as an effect called transcytosis. Also faster uptake and better diffusion, so it gets deeper into the collagen matrix, for example. Then we have very densely pegylated the surface, and we have made sure that the PEG chains only point outward, as inward-facing PEG chains are basically non-functional and are not needed. So making that very dense has an advantage that the shielding effect gets maximized. And I think one of the newer concepts in nanomedicine is this high lipid to drug ratio. Phospholipids are quite healthy actually for the liver, for example, so you can use a lot of them to formulate your drugs. And I think this is quite beneficial in a way that the reticuloendothelial system, such as liver and spleen mostly, they get sort of saturated by the loading dose. And if there is a lot of lipid and little drug, then the cleaning systems are mostly busy cleaning not that much of the injected dose, and the rest is available to tumors. To compare that against the Calyx Doxil, you see that we have a very high particle density per mil. And that basically creates a 6-fold or 600% increase in total particles injected in a therapeutic dose in humans. And also the particles are perfectly spherical, very homogeneous in polydispersity. And yeah, this is the sort of slightly elliptic form that the Calyx is known for. Regarding immunogenic induction of markers, I think it's quite interesting that Calyx, because probably the pegylation and the size, it has issues to actually being taken up by cells, and therefore induce these effects that doxorubicin is actually capable of inducing. So I think we ameliorated that significantly with Talidox. It has a greater induction of markers of the immunogenic cell death, HMGB1, for example, or also calreticulin. Generally, doxorubicin is well known to actually down-regulate PD-L1. So I think in a way, it's also quite a good immunotherapeutic. Probably one of the reasons why, like 70 years later after discovery of doxorubicin, it's still one of the most used anti-cancer drugs. But also other molecules are triggered by the use of doxorubicin. So coming to phase 1, I think it was very interesting for us to see that Talidox gave rise to a very large AUC. I think it's one of the largest AUCs I've seen for chemotherapeutics. It has an approximate serum half-life of 98 hours. We took here the 45 milligram comparison dose from the phase 1. To the study by Ranson and colleagues. Efficacy-wise, I think from all the cancers we treated in this study, breast cancer was the one that responded the best to it. But there's also other cancers that are interesting, but I'm showing here breast cancer patients that pretty much all of them had stable disease, and 3 of them fortunately had a partial response with complete elimination of target lesions in 1 patient. Given the dose that was given at this stage was 40 to 45 milligrams per 3 week per square meter. So we have to consider that these patients had multiple prior lines of chemotherapy. So I believe that these efficacy data are quite promising. But also very important is a comparison against literature values of standard chemotherapy that is currently in use. So these are compendial literature values. You see here doxorubicin, paclitaxel. I think the most used chemo these days in metastatic breast cancer and also other cancers, and Calyx. So I think Talidox did quite a good job regarding the reduction of nausea and vomiting. It still has an issue of mucositis, so skin toxicity mostly, that can be quite well managed. Especially we have given quite a high dose of 13 to 15 milligrams per week per square meter, which was a bit high and will go a bit lower for further studies. So I think the safety profile will again ameliorate. But certainly hair loss is greatly reduced. The cardiotoxicity has been also greatly reduced. Blood values such as neutropenia also ameliorated. Anemia completely absent. Neuropathy as well. Hand-foot syndrome still came up, but I think with a reduction of the dose in further studies, this will be also again much better. And then I wanted to point out an interesting economic aspect, actually, of chemotherapy these days. So in average, these are data from these two papers here below, per patient per year, there is a spending of $12,907 US in the United States for the management of chemotherapy-induced side effects. And if these side effects are severe, the cost again increases dramatically. So neuropathy, one of the most expensive adverse events. And these can all be quite heavily reduced almost to zero or to zero in some instances. And I think so there is a double win that patients have a better life quality, but also that the costs can be reduced for health insurers. So there is further studies coming up, such as this one, that we call it the SMART trial, coming from a smaller, because Talidox is a smaller nanoparticle, and stronger because it can be dosed at higher intensities, where we compare in the same patients the PK of Talidox against Calyx, and the subsequent continuous treatment with Talidox. So this is at the 6 of 14 patients, where we actually don't want to show that it's bioequivalent, but actually that the physicochemical changes of the nanoparticle generate something that I would call biosuperiority. It's not bioequivalent. It's also not bioinferior. It's, I think, something new. And there is a second study called Traction, where we'll go in efficacy and safety against the standard of care, which is in most cases paclitaxel, but also Calyx. And there is a crossover to second line for all the patients. So the rationale is that the Talidox can maintain or even surpass efficacy of comparators, and that the tolerability safety profile is better, such that the quality of life and the patient reported outcome measures are significantly better. Regulatory wise, I think it's interesting to note that Talidox in the regulation of Swissmedic is a known substance with innovation. In the EMA kingdom, it's a hybrid medicine, and in United States, it's a 505(b)(2). Also, outside of clinical trials, Talidox is now these days available in Switzerland, and we're making efforts that it's also available outside of the countries as a formula magistralis. So we do have quite a pipeline in development. We have Talidox in oncology here. In breast cancer, we're entering phase 2-3 trials now, and there's formula magistralis that is already on the market, and there's further developments such as a docetaxel compound or a tracer. And I recommend also that you attend the talk tomorrow at 10:50 by Dr. Camille Peitsch. She'll be talking about one of our most important medicines, talinurine, for the treatment of Parkinson's disease. So I thank you very much for your attention. And if you want to exchange with us about liposomes, collaborations, or partnerships, feel free to reach out to me or somebody of the company. Thank you very much.
Thanks, Stefan, for a really interesting talk and appealing clinical pipeline going forward. I think we switch gears to not only liposomes, but other type of lipid nanoparticles, and actually also surrounded by the upcoming talk of Professor Avi Schroeder. So really interesting to learn your data. And actually I saw them last week at an interesting presentation at the Bauernholz conference on enhanced uptake during the menstrual cycle. But I think you will also elaborate on pancreatic stroma degeneration, as well as also patient-specific approaches. So Avi, the floor is yours.
Thank you so much, Cristianne, and really I enjoyed the talks very much, and I'll be talking more from an academic perspective. I work at the Technion, which is one of Israel's universities. Can you hear me well? Okay, great. And really about some recent discoveries we saw actually in the use of nanotechnology or of lipid nanoparticles with regard to gender and regard to also different biological barriers that we have inside the body. And really, in the interest of time, I'll try to keep within the frame of the 15 minutes that I was given. So really, first, biological barrier, I'll say maybe I'll start from papers we all saw where we see very massive clinical accumulation of the nanoparticles in patients. However, it doesn't always translate to clinical efficacy. So if we look at papers throughout the years of imaging of the biodistribution of the nanoparticles, we see them at the tumor site or at the disease site, but we don't always get that higher accumulation. It doesn't fully translate to activity. And one of the reasons is actually collagen, which we have in different disease sites inside the body. For example, in pancreatic cancer, collagen actually shields the cancer cells from drugs penetrating in. So I once thought of collagen only as a barrier, maybe as a wall or the vault for the safe where you can't actually penetrate. But actually, studies that were conducted in the late 1980s, 1990s showed beautifully that collagen also acts almost as a glue, deactivating drugs. And I think the same is also true for our nanoparticles. And if we look at pancreatic cancer, this is a pancreatic tumor, you can actually see on the right-hand side of your screen a cut of a pancreatic tumor. All the blue regions that are inside this tissue cut are collagen, which is shielding the cancer cells and preventing from drugs to penetrate in. This work was led by Asaf Zinger, who's now faculty, a former student of my faculty today at the Technion. On the left-hand side, he actually looked at the healthy pancreases, and you can see there is collagen there. So there's some bluish stain, but however, this collagen is more a structural component rather than a shielding component. And what we asked ourselves is, could we take a liposomal formulation, load an enzyme into these liposomes and actually have this enzyme specific to degrading collagen, which is inside the collagen, inside the pancreatic tumor. More specifically, Asaf loaded collagenase type 1, which is specific for collagen type 1, and it's an enzyme that will degrade or cut the collagen matrix. And then our thought was that this would allow the nanoparticles of the liposomes to penetrate better, a drug to penetrate into the system. And indeed, when we tested this liposomal collagenase, the reason you need to load the collagenase into a liposome is that collagenase is very active, but it also deactivates very rapidly. And the liposome protects the collagenase by not activating it before it reaches the target site through different ions such as calcium. And so the collagenase was released from the liposomes. It was able to degrade the collagen matrix. And we saw this also in different collagen fibers, but very nicely when we decellularized a pancreatic tumor. You can see in insert E on the right-hand side, bottom right-hand side of your screen, we were able actually to reduce the load or the burden of collagen inside the pancreatic tumor. So these are mice that have pancreatic tumors. The liposomes with the collagenase are injected intravenously. They distribute throughout the body, and we also showed they accumulate well inside the pancreatic tumor. They get stuck by the collagenase. They open up, and actually the collagenase is capable of reducing that collagen load, creating pores that are large enough for a second treatment now of a drug-loaded nanoparticle that can treat the mice. And in fact, when we tested this in vivo, and this was published in ACS Nano just a couple of years ago, when we injected these liposomes first with a collagenase, then we treated with a liposome with a paclitaxel, a nanoparticle paclitaxel, we actually were able to improve the therapeutic outcome in treating pancreatic tumors. Today, I may have chosen a different drug, and this touches maybe on the previous talks, where a platform system actually shows a lot of the safety and efficacy of a liposomal carrier, but you can actually swap different drugs, but you do have to tune the lipid bilayer. And I think, we are in a very translational session here. One of the pressure points we need to apply more on the regulatory bodies is that for a knowledge that we already have on the biodistribution and safety of different components, I'm not sure we have to run all the safety over and over again. It just is killing very good projects before they reach the clinic. And when we weigh the benefit of the patients versus the chance that a specific lipid may actually induce an unknown side effect, I think as scientists, we can give a pretty good indication that swapping the drug, that's the main change, and not swapping one or two lipids inside the component. And if we continue the way we have, I'll say this point blank, I said this a week ago to a leader in the FDA, we're going to be preventing from the public very good drugs. And I think COVID taught us that sometimes speed is actually very important for saving lives. So that's like my bottom line. But anyway, going back to pancreatic cancer, we showed that you can actually treat here and improve the treatment by reducing the collagen load at the treatment site. So the biological barriers really play an important role in deactivating sometimes our drug delivery systems. However, when we were working on this study, something very interesting happened. We were asked during the revisions to look again at the biodistribution of the nanoparticles in the mice. And the students that were working at the time, Asaf Zinger and Maria Prolai, chose to test not only in male mice, but also in female mice. And what they saw is a very strange biodistribution pattern. They actually saw nanoparticles accumulating in this region of the body. You can notice it's a higher bright region on the right-hand mouse, which I initially thought may be the bladder. However, this was not the bladder. This was the reproductive system of the female mice, where we saw a higher accumulation of the liposomes. And what happened in the animal house was important to notice. So we had male mice and female mice actually in cages that were next to each other. And the people working in the animal house by mistake mixed the bedding of the male mice with the female mice, meaning that the female mice were exposed to the bedding of the male mice. And once female mice are exposed to the scent of a male mouse, actually this triggers an ovulation cycle or an estrous cycle in the female mice. So what happened is that we had a triggering of the ovulation, and that actually adjusted the biodistribution of the nanoparticles towards the reproductive tract of the female mice. And this study was run by Maria Prolai, who will be starting her postdoc in Harvard in a couple of weeks, and Asaf. And they really looked at it as gender nanotechnology. Do we need to engineer our nanoparticles also to the sex of the patient who will be using it? And I believe we do need to take that into account in improving our drug delivery systems. And we know that the sex or the gender of a patient actually plays a very important role when we look at many small molecule drugs. So the same can also be true also for nanoparticles. And one of the first indications we got is that you can see on the upper panel of this slide that when we looked at the different stages of the ovulation cycle, at the peak of ovulation, at that stage that's called estrus, we had several-fold higher accumulation of the nanoparticles after being injected intravenously, meaning that when you inject the nanoparticles intravenously in the mice, we actually saw at the peak of the ovulation cycle, we saw higher accumulation of the nanoparticles inside the ovaries and the uterus of the mouse. Here we were using fluorescent nanoparticles. In order to make sure that our indication was right, we actually swapped these liposomes with lipid nanoparticles that contained a messenger RNA. And we actually saw the same thing exactly, that we were able to actually inject intravenously and encode for luciferase inside the ovaries and the uterus of the mouse post-intravenous accumulation at the peak of ovulation, meaning that these nanoparticles actually traffic through the blood and accumulate in the reproductive system. The nanoparticles themselves. So what we see here is a single cut of an ovary, and these are the egg follicles, and the nanoparticles in this slide in pink actually were parked outside the egg follicles, standing at that junction. So on the surface, and the reason we have this higher accumulation seems to be actually the increased angiogenesis to the ovaries in order to support the potential of life during the peak of ovulation. So very similarly to other biological accumulation, we had the enhanced permeability and retention effect, which was mentioned by Cristianne. And now we actually see a very similar effect actually also in the ovaries, which makes sense angiogenesis through which nanoparticles can actually accumulate inside the ovaries. And here you can actually see that increase in the density of blood vessels around the different egg follicles during the peak of ovulation, which then actually triggered the first question is there a nanotechnology aspect here? Meaning, how large a particle can actually accumulate inside the ovaries. And what we saw is that there's approximately a 100 nanometer cutoff through which the nanoparticles can accumulate well into the ovaries. After which, when you go higher, 150, 200 nanometers, you actually see very little accumulation in our studies, at least. This may change from particle to particle. And also, I think the different configuration of a particle may also play a role. So we used spherical particles in this study. So how could this be leveraged? When treating ovarian cancer, we may want to treat at the peak of ovulation so that we get maximal uptake of the drug in the ovaries and to the region of the tumor during the treatment. And indeed, we saw that when we treated ovarian cancer at the peak of ovulation, we saw an improvement in the clinical outcome and preclinical outcome in the mice. However, when we treated breast cancer, so this is a breast cancer tumor, we actually saw a very distinct biodistribution and then also efficacy in the mice when we treated at the peak of ovulation or at the base of the ovulation. When we treated at the peak of ovulation, we were losing a lot of the liposomes, which were loaded with a drug, to the reproductive system, meaning that not enough drug actually reached the tumor. Instead of going to the tumor, it was going to the reproductive system, to the ovaries and the uterus. However, when we treated at the baseline of ovulation, then we actually had higher accumulation in the tumor and better preclinical results in the treatment. And we saw it very distinctly also in the biodistribution and also in the accumulation. And this was published recently in ACS Nano. I think in the interest of time, right, Cristianne, I'm up with my 15 minutes. So I'll just end this concept with a nice slide that was produced by Rafael Torres Martin de Rosales. You can actually see here a mouse which was injected liposomes which have a tracer in them. These mice actually have both a breast cancer tumor, and you can see, though, the accumulation also of these liposomes in the reproductive system. This surely was not the intention of this treatment. The treatment here was supposed to be focused at the breast cancer tumor, but it actually almost distributed evenly between the breast tumor and the ovaries and the uterus. And that, of course, can have positive or negative effects based on what we want to do. So I just want to end by saying that timing the result is important. And if I may, I'll just end with one slide of a project we've been initiating over the past years, and this is past year, sorry. And this is really of integrating artificial intelligence into the design of nanoparticles. I think in many of our labs, surely in mine, we were using, I would say, old-fashioned methods for producing the liposomes, which were based mainly on the knowledge we had in the field and our expertise. And throughout the past year, we've been gathering more and more data on the different aspects of liposomal and lipid nanoparticle configurations, loading this into this database into a smart system which learns which lipids work best with which payload if we're starting with mRNA. And actually, we were able to predict several formulations, several new molecules that can be interesting for drug delivery. So I think when we look forward, one aspect which is entering our world is synthetic biology of drug delivery. But I would say the second aspect, which is playing a very important role in many other fields, and now is also playing an important role in our world, is actually artificial intelligence, where artificial intelligence can really help the way we construct, build, and test, and also evaluate the efficacy. So thank you very much for giving me the platform today, and it's been a privilege to speak here.
Thanks, Avi. I think for me, the summary of your contribution is that a picture says always more than 1,000 words. So I think that it really shows that the imaging is so powerful, and if you can combine it with artificial intelligence, then who knows what the future will bring. So very much looking forward to anybody's else questions, but as said, please keep them till the end of this session. So we have Professor Nam Jong Cho, if I pronounce it correctly. And instead of generating new membranes, this talk will be rather on attacking membranes, more specifically by membrane-active peptides, which aim to exhibit broad-spectrum antiviral activity. And clearly this is applicable to a whole range of different viruses. So, Professor, the floor is yours.
All right, thank you very much. Can you see the slide?
Yes.
All right. Thank you very much for the this opportunity, and I'm really enjoyed the listening to the talk.
Sorry, please. Can you go in the setting display and share your?
Now it's better.
Yeah, it's good. Thank you.
All right, thank you very much. So today my talk is a little bit. I do lipid nanoparticle work, and then this is one of the platform I'm using. That lipid nanoparticle platform as the drug screening platform to actually figure it out how do you actually screening the peptide. And then this is the topic of last 10 year plus we working on the broad spectrum antiviral peptide that combating emerging viral pathogen like including the COVID. So this concept is basically what we call this lipid envelope antiviral disruption, a lead concept we published in the Nature Communication and a lot of Nature material and a lot of other journal, and that we call the Achilles heel of the emerging virus. So basically, as you see that, and then the current view of this viral infection, viral pandemic, including the COVID-19, and then actually you see that this mapping in here is only a small fraction of the virus is we identify and that we're working on, we're focusing on, which is HCV, dengue, and chikungunya, JEV, so on and so forth. But if you actually open up the mapping, there is a lot of unknown and emerging, and we're going to come again. So basically, we need to prepare unknown unknowns. And also, and then as you experience with the COVID-19 pandemic, there is a continues to mutate and evolve. Then the people thought last year, for example, and people thought we can control, but Omicron came, and then everything once again is screwed up right now in China. And then I know that in Europe, they open up, US open up right now, Singapore right now, we are open up the market, but what about China right now? They have a very severe problem. So we need to have a countermeasure for new and resistant viral threat. And then there is a plenty of opportunity evolving the strain, but the current vaccine cannot stop. So, and then there is another announcement that, you know, by the Bill Gates, and they talk about in Munich, and also a recent TED talk, we need to really prepare the next pandemic. So there is a lot of class of antiviral strategy by looking at this mapping of the viral infection mapping, and the people try to do basically looking at this pathway to stop one of the place, you know, stop the inhibiting transcription, inhibiting translation, inhibiting assembly, and so on. But what we're suggesting in here right now is a lipid envelope antiviral disruption platform that can be actually using it for the new class of drug that target the liposome below 160 nanometer, which is lipid envelope of virus below 160 nanometer size. So this whole concept or peptide is derived from actually its own virus. So I was working in the HCV before, and they have this domain called non-structural 5A protein. That from there, we drive and we engineer the peptide that sensing the curvature of the lipid envelope, and which is the amazingly working very specifically and sufficiently. So what we design is basically we take this initial amphiphilic helical part of the peptide and we design and what we call this H peptide, which this peptide is quite interesting in the sense that 2/3 is hydrophilic and 1/3 is hydrophobic. And then this non-helical peptide, basically we design as a negative control. And then now we design the new peptide, lead peptide concept, generation 3, which we reduce down the toxicity and increasing the specificity. So what this saying is basically this can target lipid envelope of the virus which is a small sized curvature, which is the size below 150 nanometer. That is the most of the flavivirus, alpha virus, and so on. For example, the specificity wise, even though it's the same virus, like vaccinia virus, which is envelope RNA virus, which size is bigger than the 100 nanometer, which does not have any efficacy. However, it only works for the virus particle below certain size. And then if we mapping up and then study how this age peptide effectively lyses the size below 150 nanometer, which is working against, including coronavirus, influenza A, Hep C, dengue, West Nile, Hep B virus. So with that, we study simulation study, mechanism study, how this peptide effectively working against the lysis the membrane. That's what we studied it. And you may think this is similar as antimicrobial peptide, which is not the case, because the range we are lysis and rupturing, which is nanomolar range of the peptide concentration, can effectively working, and it does not have any toxicity effect within the membrane cell or other cell membrane. So what we did is basically we are engineered the liposome, artificial liposome, or actual envelope virus inactivated, and then we fluorescence label such as this schematic. We can use it for the small size vesicle control or big size vesicle, or even real lipid envelope from the viral particle. And then if you coating fluorescence license, you can clearly see that the platform showing as the fluorescence intensity-based images. And with that, we actually adding the peptide and follow up how this peptide lyses. The meaning is diminish the intensity of this fluorescence. This is what we call this tethered LNP platform. That's what we control. And we study with the various of different virus particle, which is what we think is optimized for the dengue, yellow fever, JV, chikungunya, Ebola, Marburg, and so on and so forth. So effectively, it's working in vitro study-wise, working on the various of broad spectrum virus particle, and it does not showing any toxicity for the avoid the larger cell in this particular case. So this lead peptide generation 3 concept is we using the 27 amino acid, low nanomolecular IC50 value for the countermeasure for resistance COVID-19. So we designed various of different peptides that we test with the COVID-19, and then you can see that the new potent viral inhibitor that acting low nanomolar range, which is the 30 nanomolar range, and so on and so forth. And then we also study PK/PD evaluation for this particular peptide. And then you can clearly see that 25 milligram per kilogram IV administration mouse for 24 hours, you see that over 24-hour peptide concentration range between the 10 to 50 micromolar, and then efficacy range is the nanomolar range. And then with that, we actually directly study the animal toxicity study design and showing in here with the intravenous and 6 vagal dog, 3 male, 3 female. We put it up to dosage up to 45 milligram per kilogram. Right now, we can administrate up to 100 milligram per kilogram. And then you clearly see the whole data showing that the hematological results for the lead COVID were indistinguishable to the control for the day 7 to 28 days. And then also even high dosages, lead COVID-19 have a similar coagulation parameter result compared to the control. So this is a very exciting data in a sense that we can showing that it does not show any toxicity. So compare with the existing antiviral drug, these broad spectrum drugs have a favorable property, the potential effective therapy against the current and future COVID strain, not only COVID, but unknown virus. For the IC50 half-life time, safety is basically defined. And then, so I'm going to give you the whole study for this lead peptide for one of the example of a Zika, which we published in the Nature material paper at that interview with the Nature. So one of the problem statement is basically diagnostic occur when clinical symptom observed and viral infection is happen. And then in this particular case is we want to design something penetrate the BBB so that we can actually clear out the viral load in the brain, and then it potentially translatable to the other mosquito-borne virus for the co-circulating with the Zika. That was one of the study, and then what we designed basically some of the amphiphilic peptides cross the intact BBB. So this is the whole design plan for the peptide. We start with the viral envelope targeting material and that inhibits the viral spreading and therapeutic solution. That's what we measure. So because of my background at engineering and medical and in interface, so we start with the biophysical design that is including the peptide engineering, liposome rupturing, kinetic membrane curvature sensing, peptide design. Then, antiviral strategy is we check the neutralization efficacy and neuron cell protection and human cell compatibility. Therapeutic evaluation is for the clinical finding, viral load analysis, and for the animal study, that's what we did. So, and let's start with this in vitro antiviral activity-wise, protect against Zika-induced death primary neuron culture, and then inhibit against not only Zika, dengue, chikungunya, JV, yellow fever, and so on and so forth. So therapeutic index is up to 5,000 in this particular case. And then this is a quite interesting study in a sense that in vivo study, therapeutic efficacy study, conventionally the animal model design people do is basically they inject the virus particle with the drug, mixing it together, then they start the injection. What we did is basically we start the treatment after the Zika injection, and then we have the viral load enough so that we can actually check how this H peptide therapy is efficient for this particular case, starting from the day 3, and then we measure day 4 and day 7. And then showing that this lethality model, 100% lethality there, we can actually rescue 86% of the mice in this particular case. And then there is a protect against the weight loss, eye pressure changes, and so on and so forth. And then with that, the viral load, including the brain, you can clearly see that we can actually decrease the 3 log loads in serum, spleen, brain, and optical nerve in this particular case. The therapy significantly reduced the viral load in various places. And then the interesting thing is the brain. So what we study with this peptide, we actually check and study how this material or peptide influx, outflux with this penetrating BBB. That's what we did with the healthy mouse compared with the unknown BBB penetrating peptide and other material. And then you're showing that the influx rate is comparable to the known material as a dermorphin, which is known as they basically penetrate the BBB. And then, one of the reviewers saying that, hey, it might be because of the injury of the BBB by the Zika infection, which is happening in day 7, not happening in day 3. So we confirmed that BBB was still intact in day 3. And then its administration, antiviral therapy start, and the viral load in brain were already detectable. So this therapy is proven that protect against the BBB injury. Untreated group exhibit increasing the BBB permeability. So this is a very, very exciting result. And this real concept with the new design, you can actually see the EC50 very consistent with the various of lipid envelope virus that including Zika, different serotype of dengue, yellow fever, JV, chikungunya, and so on and so forth. So this can be the potentially future solution, and then we're developing CMC right now, pre-IND, entering to this peptide therapy that can, in this particular case, inhibit Zika infection. We're working with the COVID-19 right now. This might also address the other Zika-related medical complication, which is including the viral persistence in tissue and then other problem. And this motivates the new antiviral strategy treating the mosquito-borne viral infection and potentially future pandemic. Thank you very much for the listening my talk, and then we will take the question. Beforehand, we developed as a Luca AI said, because a lot of people working in the LNP right now, we developed the whole new way of assemble the lipid nanoparticle, and then what we call this Luca cycle, further than the microfluidic or extrusion method, and we are able to basically assemble the LNP with a different composition and the various of different sizes. So stay tuned. This work going to be published very soon, and with a very exciting development from the Luca ASL. Thank you very much.
Thank you, Noam. A really interesting talk, and particularly I find myself the absence of any adverse events of this new approach very appealing. So very much look forward to near future clinical trials. The last talk of this session will be by Dr. Alina Sigaiva. And I think we have already spoken quite a bit about patients, about particularly also patient-specific. And I think what you developed, Alina, is a technique that can even monitor cell-specific changes. So really to see intracellular changes about temperature, pH, and recently even also free radical production. Very much looking for your talk, and the floor is yours.
Thank you very much, Cristianne, for your kind introduction. I am very excited to be here today, albeit online, and to be in this session where we've heard already a lot of amazing talks on the new therapeutic strategies. And indeed, my talk will be a little bit different, as we will be dealing not with eliciting some sort of a response from the cells, but rather monitoring that response using a novel imaging technique, namely nanodiamond magnetometry. Here is the brief overview of my talk today. I will start with some basic introduction on the technique and on the possible applications of it. Then I will move on to 3 case studies, so to say, where I will try to illustrate the potential of this approach and its versatility. And then there will be some closing remarks on the future of this technology and how we see it developing further. As all of you will probably agree to me, each and every patient is different, each and every patient is unique. Within each patient, we also have a wide range of different tissues and organs, each of which is composed of a plethora of different cell types and different cell states. And I think that you will agree with me that understanding the unique biology of the patient and the cellular diversity that underlies this unique biology is what actually allows for highly targeted interventions. It becomes especially important in cases like cancer treatment. I was asked to share the presentation in full screen. I will do this now. I hope this works better now. It becomes especially important in cases like cancer, where a very small subpopulation that escapes the treatment can give rise to the recurrent cancer and can eventually lead to the death of the patient.
Can I cut the presentation? It's
yes,
still not in full screen.
Let me then pause or stop share and reshare. Does this work now? Now it's loading, you see.
We receive nothing. No. Okay, we see nothing. We only see that you started the screen sharing, but we don't see your presentation.
Alina, do you hear us or are you frozen? seems that Alina is frozen.
Okay, so we're gonna. Where? Can you? Yes. Okay, you're
Yes.
reconnected. We're gonna
Yes.
try it, and then
share screen again, and I will start it in full screen and let me know if this is working now.
yes, it's good. Thank you.
Yay! Perfect, thanks. So as I have mentioned at the beginning of my talk, we're using a specific kind of nanoparticles, fluorescent nanodiamonds, to get an insight really into this single cell physiology and into the responses of these single cells to the interventions that we might be interested in. Fluorescent nanodiamonds are nanodiamonds with a defect of crystal structure. They're really small, ranging from 40 to 150 nanometers in size. They are very biocompatible, both when we look at them in vitro in the cell cultures and in vivo in live organisms up to mammals. They are fluorescent and at the same time not susceptible to photobleaching or blinking, which allows one to really track that same particle and that same cell that is carrying it for a very long term. They're also visible in electron microscopy, which is convenient for some follow-up studies. But most importantly, they have unique properties of those fluorescent effects. And these properties can be used for quantum sensing, which is the foundation of our method, nanodiamond magnetometry. As I said, the nanodiamonds are fluorescent due to their defects. They're called nitrogen vacancy centers. And in this case, one atom of carbon is replaced by nitrogen, and the position next to it is vacant. This whole thing is fluorescent. What's more important, its energy states are arranged in such a way that its fluorescence can be different in its intensity. We can use this to our advantage. We can use the laser pulse to prime the nanodiamond to make its fluorescence as high as possible. And then if we give the particle a certain time to relax, this whole quantum system to relax, then the fluorescence will return to a less bright state. This is exactly what we're doing. So we are pumping the nanodiamonds into a very bright state and then give them a little bit of time to relax and measure their fluorescence. If we repeat these processes with different dark times and then plot the fluorescence against those dark times, we can get a plot like this. You see that the longer we give the system to relax, the lower the fluorescence becomes. And this curve is characteristic of the nanodiamond, but also of its immediate environment. It can be shifted to the left, giving us a shorter T1, the constant that describes the rate of this relaxation, or it can be shifted to the right, meaning that the T1 becomes longer and the decay happens slower. In essence, what we're doing here, and there is a reason why we're using the T1 constant, is magnetic resonance imaging, but we don't need any magnets for it, and our resolution is substantially better than that of the conventional MRI. Since our resolution is that good, it also means that we start seeing other paramagnetic species, so we don't need external paramagnetic markers or dyes such as gadolinium. And one of those paramagnetic species that we become sensitive to is free radicals. You probably know that free radicals play very important roles in a wide range of processes. Sometimes we want to have less of them, for example, to curb the inflammation. Sometimes we want, on the other hand, to ramp up the production of free radicals to kill the cells that we don't want to have in our body. And it is therefore very important for us to know where, when, and in which quantities those species are produced. However, the methods that we have today are often lacking sensitivity and specificity for those, which poses quite a big problem. As these species are paramagnetic, they will influence this process of relaxation, and with a higher concentration of free radicals and faster relaxation of T1, the T1 time will shorten. With lower concentration of free radicals caused, for instance, by us adding some antioxidants and radical scavengers to the sample, we will see longer T1s corresponding to lower concentration of radicals. It means that within one cell, within a biological sample, we can use our setup to really track the dynamic behavior of free radicals and the free radical load on a single cell level in pretty much real time. Let me move on to some examples of how this can be used. And the first one, in the first case, we were studying the production of nitric oxide, which is an important signaling molecule and also a radical in macrophages. We had the nanodiamonds inside our macrophage cells that were expressing NOS, nitric oxide synthase, which produces nitric oxide. This process can be inhibited by the chemical inhibitors, and we would expect to see lower concentration of radicals, decreased magnetic signal, and longer T1. And this is exactly what we see with our magnetometry. You see that upon the addition of the inhibitor, the blue line, the T1 values grow over time. You can also see that the error bars are rather large, which corresponds to the differential response of the different cells within the sample. And this is also something that we can see very, very nicely with our method, and which is largely inaccessible to other techniques used to detect free radicals. We can also perform the measurements outside of the cells. In this next experiment, we had the diamonds in the cell, next to the cell, and also next to the cell, but a bit further away. And we did the same thing. And here you can see that, yes, we detect the increase in T1 inside the cell, also close to the cell. And interestingly, this response comes faster than the one inside the cells. And if we move away from the cells further than 5 micrometers, there is basically no change in the T1 values. It corresponds nicely with the other data on the diffusion of the nitric oxide in the biological samples. And basically, the reason why we see this response faster on the cell membrane, this is the distance from the cell membrane, and this is the time, is because a lot of those NOS, nitric oxide producing enzymes, are actually located on the cellular membrane in the J774 macrophages. So when we introduce the inhibitor to the medium, those will be the molecules that will first see the inhibitor and that will first stop the production of nitric oxide, eliciting the response there. The next case that I would like to talk about is us looking into the free radical load in the HT29 colon adenocarcinoma cells. This is a very interesting cell line because it can be, with a very simple treatment, redifferentiated into more functional goblet-like cells, or it can be propagated as tumor cells, giving us a very nice model to see whether there is a difference in free radical load between the cancerous cells and the more differentiated cells. In previous studies, such a difference has already been found. Domokos in 2009 showed with electron spin resonance a technique that is very precise, very selective for free radicals, but also requires a large number of cells, a large amount of sample, that these redifferentiated cells have relatively high radical load and the cancer cells have relatively low free radical load. We wanted to see if this is indeed the case, and we wanted to also not just look at the end stage of redifferentiated versus dedifferentiated ones, but also at the process of redifferentiation and to see when these differences emerge. Luckily for us, we can indeed redifferentiate the cells and still have FNDs inside. So in panels B, you see vilin, the marker of differentiation, and here in red, you see nanodiamond particles in the cell clusters. If we perform our measurements, we see that indeed by day 10 and 13, when redifferentiation occurs, the control cells without the butyrate have longer T1s, so lower free radical load, and the butyrate-treated redifferentiated ones show lower T1s, so higher free radical load. So the findings were confirmed. However, these differences emerge not because butyrate causes lower T1s and higher free radical load, but because the cells that are left to propagate without any treatment show progressively longer T1s, progressively lower free radical load. It is an interesting finding because it might mean that it is not specifically the differentiation state that gives difference to these, gives rise to these differences in radical load, but maybe the number of the cells in the sample or the interaction of those cells with each other. What is also interesting is in the non-treated sample, you see again the larger error bars, meaning that there is a range of T1 values, a range of free radical load within the same cell population. So maybe there are several subpopulations of cells, and it might have some clinical significance, for example, if those subpopulations would have differential response to drugs, to different treatment. And then finally, I would like to talk about the most clinical of our projects, where we were looking at the response of the primary bronchial epithelial cells from either healthy donors or patients with COPD to the cigarette smoke extract. It is known that cigarette smoke is bad for your health and that bronchial epithelial cells suffer from the cigarette smoke toxicity when exposed to it. However, all the responses that you can record, you can only record some hours and days after the exposure has happened. Here, for example, we used the standard fluorescent assay for a reactive oxygen species, and you see that within the first 2 hours, you don't really see any difference upon adding the cigarette smoke extract from the negative control, as opposed to, for example, the positive control. What we did is we performed our T1 measurements in those cells. And in case of healthy donors, you see that if we increase the concentration of cigarette smoke extract, the average T1 time decreases, meaning larger free radical load. This is concentration dependent, and it happens already within the first 20 minutes of incubation, which is not something that we can detect by the conventional fluorescent assays. Moreover, if we look at the unchallenged cells from the COPD patients, then they also have somewhat higher radical load when not exposed to cigarette smoke extract, although these differences are not statistically significant. And when we add already a very small concentration of CSC, we see substantial drop in the T1, substantial increase in free radical load at the concentration that, again, doesn't cause any response in the cells from healthy donors. Then if we keep increasing the cigarette smoke extract concentration, these differences become less obvious. So we see that the health status of the donor indeed affects the response of the cells to the external stressor. And we can see it very nicely and at the same time very quickly with our technology. To sum up, our method can indeed be performed on a single cell level. It gives you information on the dynamics of the free radical production inside a living mammalian cell on a very small scale. It can reflect the spatial differences in the free radical production in the sample. And these nanodiamond probes are very versatile and the sample preparation is very easy. The surface of the probes can also be functionalized with either targeting moieties or the drugs. My colleagues have, for example, recently published a paper where the diamonds were conjugated with virus particles and the changes in free radical load were tracked during the viral infection of the mammalian cells. And we can use this technology to highlight the differences in free radical production on different levels, from subcellular, the sources of this induced or reduced radical production, on the population level, highlighting the subpopulations, the rare cell types, and the cellular fate. On the organ and tissue level, because we can also look at the interaction of different cell types and how one cell type reacts to the changes in the other cell type. And finally, on the organism level, looking at the samples from donors with different health status and seeing how they respond to external triggers, to external stressors, or to the treatment. With that, I would like to finish my talk and thank all my co-authors and collaborators, as well as the funding bodies, for this opportunity. Thank you again.
Thanks, Alina, for the last contribution. Really cool to see the different effects of, well, not only cancer cells communication, but also smoke. And I think such an alternative tool was always very valuable. I think we now need to switch to the chat, for which the link is provided in the comment in the chat function. So I'll see you there in 2 seconds.